Good afternoon, everyone, and thank you so much for joining us. My name is June Black, and it's my pleasure to introduce you to Geraldine Ondrizek, who will be speaking about her installation, Shades of White. This is the second program that we've offered in conjunction with the exhibition. So if you missed the first talk by Alexandra Stern and would like to learn more about the history of eugenics in this country, you can find that talk on the JSMA's YouTube channel. Today, after Jerry finishes her talk, I hope you'll join us in the Artist Project space to see the work in person. And there will also be a reception in the Papay Reception Hall just across the lobby, so please stay for that as well. Just a little bit about Jerry before she comes up to speak. She received her BFA from Carnegie-Mellon University and her MFA from the University of Washington. She's been a professor of art at Reed College in Portland since 1994. She's received 2 career opportunity grants from the Ford Family Foundation, funding from the Houston Foundation and funding from UNESCO as well. This year, she was awarded one of 3 Hallie Ford Fellowships, a very prestigious award for artists in Oregon. And we have another Hallie Ford Fellow with us this afternoon. I'm not sure where Tanaz is, but. So I was introduced to Jerry's work about 2 years ago now by a former colleague of mine, Jessie DiTilio. Jessie and I at that time were managing the Artist Project space here at the museum, which was generously funded by the Ford Family Foundation, to help us exhibit works by mid-career Oregon artists. As we were laying out the schedule for the next several years, we called and visited a number of Oregon artists to learn more about their work and talk about the possibility of exhibiting at the museum. So it was during that process that Jessie told me about Jerry's work, and I was immediately drawn not only to her aesthetic, but to the intense subjects that she was able to distill in such a beautiful way. So I'll let Jerry explain a little bit more about how this project came to be at the museum, but I wanted to say that she has been one of the most delightful artists that I've had the pleasure of working with during my career, so please join me in welcoming Jerry.
Thanks, June. This has been a very long and complicated piece to make, and I have many people to thank that helped me along the way. This was a 2-year project in the making, and first it begins with Dr. Alexandra Stern contacting me to use my work for a project she was working on. She sent me her books, and I'll speak a bit about those later, but it really opened the door for me to understand the history of eugenics, and I believe her books have also made others willing to speak about racism in this country in a new way. Excuse me. I was invited to do this exhibition soon after I met Alex, and I'd love to thank June Black and Jessie DiTilio, the former assistant curator, who invited me to do this work for the project space. As an artist who mainly works with installations, environments, social and political issues, it is not easy to find those who are interested and or willing to let me make a work. June has been an amazing partner in putting this exhibition together, and her interest and knowledge of the work has been impressive. I'd also like to thank the director of the museum, Jill Hartz, for her support of this show and artists such as myself doing experimental projects. Finally, I'd like to thank my Reed students who helped me along the way, Marissa Barone, and mostly CJ Davis, who helped me with every step of this project all summer long, and it was hard, and former student Emily Johnson, who helped me do the publication. They were vital to my process. And I will mention my students throughout the talk today because they've always been vital to my process. I begin with this film still of gastralization, because this is the formation of a single cell into a fetal form, and it is the most vital moment in the life of any creature. The ability for this cluster of cells to survive, and how long and how well a living being survives, is determined by 2 factors, genetics and the environment. With the hand of man in the mix, the environment and now the genetics can vastly change. My choice as an artist to use the history of science and current scientific research is both personal and political. My works are in dialogue with current medical history and connected to genetics and eugenics, as you've seen in the piece Shades of White. But this concept isn't new to my work. This particular piece dealing with skin color certainly is, but looking back at all of my work, I can see that most of it has been dealing with categorization and classification, and I should also say very often it's found or ready-made images made by scientists or the formulation made by scientists, the study. So to begin with, I should first credit this man in defining eugenics. Sir Francis Galton, the cousin of Darwin, who lived between 1822 and 1911, coined the term genetics, or good genetics, eu meaning good, and it is his work in England that set the pace for the United States. I'll read a small excerpt from his paper, Eugenics, Its Definition, Scope, and Its Aims, from July 1904. Eugenics is the science which deals with all influences that improve the inborn qualities of race, also with those that develop them to the utmost advantage. Let us for a moment suppose that the practice of eugenics should hereafter raise the average quality of our nation. The race as a whole would be less foolish, less frivolous, less excitable, and we should be better fitted to fulfill our vast imperial opportunities. And I bring you Christine Borland's work, a British artist that I think sums up the vast imperial opportunities. This is a piece she did in commemoration of skulls found in a museum in Liverpool. They are made with British china. In the 18th century, Liverpool was one of the places where this china was produced. And they are from South America via the slave trade. So these skulls were found in the boats. And she really wanted to make a point about the vast opportunities of the nation. So eugenics comes to the United States via both Galton and von Luschen's skin color chart, and comes first, and probably the longest time and most important space, is the Cold Harbor Springs Genetic Laboratory in Long Island. And beginning in 1905, they opened their doors to genetic counseling, or eugenics counseling, where they would do family trees to show how to have better breeding. This was a story in the New York Times on October 13th about a museum that they have created in Manhattan, commemorating the eugenics record office. They've opened up a lot of those records for people to explore. And in many states in the United States, the eugenics practices continued pretty solidly through the 1930s, and it's actually many of our experimentations that established what the National Socialists would use. And in some states, like Oregon, it continued well into the 1980s. So my own work in graduate school first started with working in a greenhouse, a botany greenhouse, collecting plants, learning the Linnaean classification system, and translating those names from Greek and Latin so that I could understand where they came from. And what I learned was how both derogatory and colonialist even the naming of plants were. And I would say much of my work is preoccupied with hothouse flowers and genetic anomalies. I created for my graduate thesis exhibition a collector's chamber, or a theater of knowledge, that directly referenced the hierarchies of knowledge and power. Systems of identification and order placed upon life became central to my work. Social Darwinism was present everywhere to me, and it has been in existence in the naming and collecting of all living organisms since the Linnaean classification system, but arguably this goes much deeper in the history of humanity. As Stuart Hall points out in the book, The West and the Rest, visual images have played a central role in describing these oddities or anomalies, and by using exaggeration of their features. Because of time, I can't possibly show you the history of this, the history of the art images or the scientific images, but I found a few that might get the point across. The first of which is Charles Le Brun's 1690 physiognomic heads. The goal was to judge the character of a person based on the facial characteristics corresponded to animals. The angle of the nose, the eyebrows, etc., the chin. In this case, it was the eagle-nosed, who was particularly untrustworthy. And these were done for the court of Louis XIV. We could go on and talk about Napoleon's 1798 French army of the Orient that was accompanied by the Committee of Arts and Sciences, or Daumier's sketchbooks of caricatures or Degas' pictures of prison inmates. But I think probably the most telling in the way in which we used art and imagery as a people was the 1937 Degenerate Art Exhibition staged by the National Socialists that compared artists' work to images of those who were physically and mentally handicapped. Here we have Modigliani, this is a Modigliani here, and a Carl Schmidt-Rodolph next to a handicapped person. And it was, the term degenerate was adopted by the National Socialist regime as part of its campaign against modern art. But as we know, it was also against people with any kind of disabilities. Many works branded as such by the Nazis were seized from museums and private collections. There was a 3-year exhibition that crossed Germany and Austria, and the surviving works were just shown this summer at the Neue Gallery in New York, and it was a stunning exhibition of what was collected and how it was compared. It was rather moving. In my own work, though I had learned this information while in graduate school studying hierarchies, genetics, eugenics, and differentiations, I continued to work within laboratories, and another exhibition experimentation that I began to engage with was to actually grow things in the laboratories. And in this case, this is white willow, which is the origins of aspirin, but I was thinking a lot about how we can force life and grow life and under artificial lights and circumstances. And this is at the same time that cloning, genetic modifications are beginning to be talked about, and the first test tube babies are born and begin to grow. My next body of work took a very serious turn. I began to think about magnification, and this is actually a picture of Robert Hooke's microscope. And the microscope, which the invention of the optical lens, actually, particularly the microscope, first invented by Anton van Leeuwenhoek of Holland, was subsequently by Robert Hooke in 1635, opened up a whole new way of seeing and then knowing. And it was my own interest in looking through microscopes while in the laboratories in graduate school that started me on this track, and at first I was looking just at plant cellular formation. But when I lost a child to a genetic anomaly, that observation profoundly changed, and it became both personal and political to look at cellular structure and genetic makeup. And I moved abruptly from plants to human beings. And this is the gestation of a cell, a single cell. I chose to use cloth, staining cloth, sewing into it, because the domestic forms of linen invoke the feminine and feminist ideas. Scientific experimentation is seen in the realm of the male, and sewing in the realm of the female, and the two hardly ever crossed over. Though what I did find was that many of those who were illustrating the cells in these first books of gestation were often females, as is the case here, which is a table runner. And this is the fertilization and the cleavage of the archaeist egg made by a woman named Van Bergen in 1883. So this is actually all of the stages of gestation. I took her drawings and actually letterpress printed them onto cloth and then hand embroidered them. And there's maybe hundreds of these that I did. And at the time, I was feeling a little bit like a test tube mother, became pregnant again, and this is the chromosome count tests that we had done of our son, and I made a series called Torah binders that have his chromosomes embroidered into them. So, in 2003, I had the opportunity to live and work in London at Gasworks, and while there, I was invited to the Wellcome Trust, which is an institute that holds is a research institute and also holds an archive of the history of eugenics and genetics. This was an amazing opportunity, and while there, I was able to access research that had recently been done on what the RNA print looks like of someone with mental illness and someone with cancer. So my studio became this space where I was determined to repair or erase out all of the markers that would be the negatives. So I strung this like a hospital curtain in my studio, and our studios at Gasworks were also public. So the university students from Goldsmith, Slade, they came through fairly often, and we had to be there to talk about our work. So it was a great opportunity. I could just be in the room and keep working while they were there. So I spent about the remainder of my time, the first part of the time was, of course, making the piece and then hanging it, et cetera, sewing this and having dialogue with people who came through. And at the time, people weren't thinking so much that genetic testing was going to be part of our health, but it certainly has become so. It also made me begin to think about labor because it was long and labor intensive to do this kind of sewing. And I began to think about sweatshops. This is a picture of the sweatshop, the Triangle Company, where the shirt-waist coat fire happened in New York City. So production tables such as this have been very common throughout the centuries. In my next work, I was given an RNA gel that from a student of mine who was working at the Center for Disease Control that showed the herpes B virus, which is the only virus that crosses between a pregnant mother and her child. And so this kind of labor of fixing this situation, and quite frankly, the students telling me about her first job doing this and how compelling it was, made me think of making this long table. This was originally made for the 9 Gallery in Portland, and it had a very tall, narrow space, so the piece fit into there. I was interested in making a table like that was used in Southeast Asia for embroidery production, and they would have the spools of thread above their heads and be able to pull them down and continue to sew. And I rather love this image because Irini is Muslim and Rachel is Jewish and Jessica was Christian, and it also brought together this whole dialogue about our lives and race and culture that intersects a lot with my work, but particularly in a personal way here. And certainly the idea of this piece was to relinquish some of the authorship and have other people sew on it. The piece traveled to New York and Whitman College, and many people have sewn onto the table. And in a sense, thinking about the labor that biologists go through right now, thinking about how can we repair, what does this look like, and how much should we repair. This is a DNA sequencer, also at the Wellcome Trust, and they did have it running. I haven't been back since 2009. I was fascinated by this machine, and at the time, DNA fingerprints were fairly common for identification. So I was interested in DNA fingerprints, and one of my students, Ben Duncan, was in my sculpture classes and doing biological research. We got a grant, and I hired him over the summer to make the DNA fingerprints of my husband's family, all of the males in his family. And at the time, the Patriot Act had been passed, and being married to an Arab American, we paid attention to this idea that the fingerprints of Arab Americans could be taken, and why, and what kind of legal use they would have in the future. This is an installation at the Portland building, which is in downtown Portland, and it's across from the FBI headquarters, so I thought it would be interesting to place the DNA fingerprints in front of the FBI headquarters to have a dialogue with them about that. What was really profound to me about the piece was that my sons, his uncles, and his grandfather had these markers that lined up in the end. So I also structured this not only for that particular space, but it's made like a rug loom, an Arabic floor rug loom. And you can see the strings shoot through the piece. So I'd been working with ideas of genetic history, and this piece really came from, again, doing some reading about the current genetic research that was going on in 2004, 2005, and the research of Luca Cavoli Spanoi and Spencer Wells in Italy first, and then at Stanford, came across to me. And what's interesting about it is that they tracked the Y chromosome, or the male Y chromosome, from Uganda and watched exactly, were able to trace exactly where it went. And perhaps you know about this, there was also a PBS special about tracing the Y chromosome. I contacted them and asked if I could use their research for a work of art, and they allowed me to do so. This is more of a physical metaphoric piece. It was designed for the Oregon College of Art and Craft. It's a atrium-like space, and so it's white Muslim panels that are layered up, and the very top has the genetic code, M168, which is the origin. If you can see, M168 is the first marker on every male's Y chromosome. So if you come from the Middle East or the Northern Europe or the Mediterranean, you probably only have 3 markers if your family has not moved around. And you can really see where your family has moved to and from. So I was trying to make a physical visual analogy of this movement and space. And you could walk into the piece and see through it, which was the top of, was M168. In addition to this, I made a series of books that lined the walls that were very much like prayer books, and lining a wall like a prayer wall. And the books are cut with a concentric circle die in them. The entire genetic code is printed into the book, and each chapter or each different level of circle is printed with the different genetic marker of different layers in the world. So all male genetic markers are in every one of the books. So this is probably my most abstract piece to date. In 2007, I began to work with the technology of cell recording with Andrew Pelling, a biophysicist at UCLA. And in 2008, I was invited to CAMEC, the Center for Art and Technology in Marnay-sur-Seine, France. And I was interested in using these cell sounds because the residency takes place in a monastery. Monasteries have been designed to have incredible acoustics. In addition, Robert Hooke, when he first looked through a microscope, he called a cell because of monastery cells. So I was paralleling that as well. So my first attempt at the piece was at Marnay-sur-Seine. And these cells that I was accessing, I'll have you listen to an auditory piece in a second. I embedded the microphone or the speakers into handmade paper. I made walls of paper that are 8 feet, and that was kind of a fun invention that my students and I came up with, of how to make paper that large. The cells were recorded by Andrew Pelling, using a biophysicist, using an atomic force microscope, an AFM microscope, which has a very fine tip to feel the actual cell, and in the same way a needle finds the pattern in a vinyl record. Pelling has found that cells with cancer or other diseases give off very low strained frequencies, while healthy cells give a more even sound. Viewers encountered this work were asked to step inside the space and hear the cells and feel them through the tiny speakers. So as you probably know, speakers are made with paper, and the paper vibrates, and that helps the sound. So I was trying to also make essentially rooms that were speakers. This is an illustration of what's going on that Andrew sent to me so that we could further explain. This is another picture. And at the residency, it was a very fun residency to do. There was a filmmaker and a dancer and a poet, so we had fun making a kind of experimental film with the sound one evening. This is the dancer. During that same year, I began to work with Steve Black, a colleague at Reed College who studies developmental biology and zoology. And this is an image, as I started with a blastiocyst, a multiple cell embryo, It was taken with an optics microfiber digital camera mounted onto Olympus stereo microscopes, and the film was made with Astro imaging software. Steve allowed me to use some of his raw films, and then he gave me a setup with the spider eggs. These are spider eggs. And we shot over 200 hours of these spider eggs developing. What I was interested in doing, I'll show you the film in a minute, was to edit the film so that this repeats over and over, so you never see the actual full gestation, but always see endless potential. And in that rhythm, you see some that live, some that die, and others that are mutated as they go along. So sort of the flux of life itself. So if I can remember how to do, whoops, that's not it. Sorry. Command tab. It's not working. I'm trying to get the film. All right. So there's a film behind there. So I could just escape this. And the film is no longer there. The film is on the, it's right behind there, I think. There it is. There's the film.
This one?
No, it was right there. Great. We're not seeing it, but we're hearing it.
Okay.
There we go. Yeah, we want that. Oh, is it not going on there?
Yeah, you're sweet.
So he told me I would need to turn the volume up. So what I did was the sound wall piece was one piece, and the cellular film and film stills was another piece for an exhibition at the Portland Art Museum. I put the two together to make a film with sound and try to orchestrate some of the sounds with the dividing of the cell in its beginning of formation. So this body of work spawned a lot of other work, but what was interesting is I showed it again in Florida, at Florida International University, and when we were curating that exhibition, I pulled out books I had made 10 years earlier, and I'll show you in a minute. I had done books of the same sort of gestation 10 years earlier before I had access to this kind of technology. So that was pretty exciting to realize. Drag that back and put it on pause. We could watch this for a long time. It's sort of like a Brian Eno musical piece my husband was talking about earlier. He can give you a whole outline of who Brian Eno is later. If we listen to this or watch the film, you would see and hear that there's a collapse of the structure, and the sounds change abruptly. The sounds of cells that are healthy, or what we were just listening to, the sounds of cells that have cancer are like sandpaper rubbing. So it's a very clear auditory sensation, and I was really fascinated by the visual and physical that go on in scientific labs. And working with the students was really, really fun. Sorry I don't have time to mention all of the characters that helped with that, but that was pretty exciting. This is an exhibition at the Portland Art Museum in 2011. They invited me to do a piece in the Apex space, and I recreated the cellular and sound wall, and also made a piece that they commissioned me to make for the front room of the space. And this piece is called Case Study. At the time, I had begun work with the University of Washington Department of Medical Genetics, and they allowed me to access some of their research. This is another image of the sound wall piece, the interior and the walls. And case studies which used the National Center for Biological Information's database. I marked every disease that all extended family members had and made a case study of my entire family and relations and what kinds of genetic anomalies or qualities one might find, blue eyes, red hair, et cetera, in addition to other sorts of things that might not be as favorable. But it was really this moment of being able to work with Dr. Arno Matofsky and Robin Bennett in particular, and Peter Bauer, actually a Reed grad at the University of Washington. They allowed me into their laboratories. I have so much imagery and information from them. Gave me articles to read. They asked me to do a piece to commemorate 50 years of medical genetics. The University of Washington is the first school in the US to have a Department of Medical Genetics, which is very different from just studying genetics. It's genetics for human beings, and they're really trying to interface with doctors. And Arno Matofsky, who started this department, is a Holocaust survivor. He was on the SS St. Louis. So when doing the piece, I was really taken by his own life story, and we had a lot of really amazing conversations. We chose together, I showed him a lot of ideas, a lot of work. He really didn't want a representation of him or his research, but all of the people who studied with him and the sorts of things that happened. And he was most proud of Mary Catherine King, who discovered the breast cancer and ovarian cancer gene. So we chose chromosome 17 as the main piece, the main image for this piece. It's a large case at the University of Washington, right next to the tea room at the front lobby, if you're ever there. It's layers of silk and cotton dyed and then hand embroidered with all of the genetic markers. So it's very tiny embroidery. The front plate of the glass is etched with images, and they depict the history of genetics. So here's a family tree, cellular division, et cetera, and then terms that are most often used. And Robin Bennett, who's the head of genetic counseling there, who's been a partner and supporter, often takes her clients past this wall, perhaps on their way to tea, but also to maybe explain this is what your test is going to look like, and we're going to be able to see, and these markers are different levels because of different levels of protein, so that she can actually have something to show them that's fairly big and fairly informative. This, of course, is a computer-generated image, right? And so I was trying to also take these pieces of data that are mincing our humanity and turn them back into humanity. This is Arno, who was, as I said, very interesting to work with and inspired, I believe, this body of work, really. I thought a lot about his own life story, because when he and I met, he is from, my family's from Czechoslovakia. He's from a small German town next to Czechoslovakia, and we both speak German, so we had an interesting conversation, but he said the reason he became a medical geneticist was because the first geneticists were eugenicists, and they got it all wrong. So he was determined to do something very different with that history. So this spawned another body of work that I most closely worked with Robin Bennett on. This is an exhibition at the Kirkland Arts Center, and it's a Syntony map. It's a colored stripe array that compares gene sequences between species to elucidate the evolution of chromosomes. So it shows what kinds of markers are changing or what is the anomaly within it. So the way in which this is used is that chromosome anomalies can be detected with this test through when you can see structural alterations or different sizes, and they're associated with various diseases. So for example, it is known that on chromosome 19, the gene implicated in leukemia is there, and if it's a large marker, there's more of a propensity. This is a really new science. It's a really new way of thinking about our bodies, and Robin really struggles with how to talk about and how to teach this. And she did use this piece on several occasions, but also did a live genetic counseling session that was on television. It was kind of like an odd thing, but and did take a lot of people there as well to see the exhibition. In addition to this piece being at the Kirkland Art Museum, it also went to the University of Houston and NASA, and the University of Miami and Florida International University. So it's traveled a bit, and with each location, we've had various panels with scientists, cancer survivors. A few dancers have choreographed some pieces. But I think the thing I'm most proud of with this piece is that Robin convinced me that we should make an edition of 10 of each of these panels into scarves, and I did bring one today. And we sold those to raise money for the Genetic Medicine Clinic for those who can't afford to have genetic testing done. And if you have the propensity for something like ovarian cancer or prostate cancer, you might actually want to have the testing done because you'd want to know. So there's a pool of money that has been created from that possibility. The actual exhibition, I made cases for each of them to be rolled up and be stored, and Reed College bought the piece, which is really quite sweet. The Oregon Arts Commission helped to fund that, but it will be displayed in the cases rather than its other format. This is an image of what the piece cellular looked like in Florida. When we designed the piece, we made a kind of cyclorama. So the film is on various stages, and you could see it unfolding all around you. And I talked about the books I had made. I made about 20 books on cellular formation, and we showed a group of them at Florida International. This is one that MIT just bought, so I was very excited about that. It was really an early lithograph series. I had never done lithographs, and this is a hand-drawn study of the cells when I was looking through microscopes. So that brings me to this piece. And it's a lot of stuff to give you all at once here. So my new work, I'm representing, as you've all seen in the literature, the Gates skin color chart to a degree. The work is based on the research of Dr. Alexander Stern, a medical historian at the University of Michigan. Stern's research in the history of eugenics and its genetic and racial discrimination as practiced in the United States from 1900 to 1987. Stern's books, Eugenic Nations, Fault and Frontiers of Better Breeding in America, 2005, and Telling Genes, The Story of Genetic Counseling in America, 2012, explore racially discriminating practices such as forced sterilization of individuals supported at the public's expense, including in children's orphanages. So that's what really was my first take on all of this, was how sad, incredibly sad it was that children were tested in orphanages to see what their skin color was. And it is this image in her book that drew me to begin to think about a way to talk about this. And certainly, talking about race is a really difficult thing, and so I began to research what actually makes up our skin color and did a little mini course on my own on the chemistry of phenylalanine, eumelanin, and melanin's levels, and learned that they were actually colors, not black or white. In fact, none of us are black or white. The Gates skin color chart is based on the Van Luschen skin color chart that was first made in 1860 in Austria. And the Van Luschen certainly was used by the National Socialists, and Gates, being a eugenicist in America, decided that he could make a better one and just pare it down to 9 different shades. So what would happen, as you can see here, is that a child who was up for adoption would be placed in a testing area so that they could decide which color family they should be with or what shade of skin they were, so they were properly placed. And to some degree, you know, it could be a good thing, but it did end up that children, for the most part, those who adopt are white upper middle class families, and those who are available for adoption were not. So there was a lot of discrepancy. This is a Van Lushen skin color chart, and it was actually made with wax. They're glass slides that he would mix the colors of skin, the skin colors with wax, and put them into these slides. He traveled all over the world to do this work, to figure out the different shades. And for a long time, people used it, but in the 1930s, they began to think that this may be not quite perfect. So we had our Gates came in and did a better job by 1940. So in this work, I was using organic dyes and silks and a variety of silks, all of which I accessed from a woman in Portland who sells silk from the Silk Road. So they're from China, they're from Uzbekistan, they're different types of silk, they're mostly hand spun. And the dyes that I use to make the work are from all over the world as well. Brazil wood, fuchsia wood, Brazil wood from Brazil, fuchsia wood from Barbados. So there's really an interesting track of where each one of those come from. And then using steel, I was interested in using steel for a couple of reasons. It's a sign of industrialization, it's clean, clinical, and I also really wanted very much to take this very feminine way of working. I work with cloth, sometimes have been called, a textile artist, put into kind of categories. And I also wanted this work to have a kind of seriousness. So putting those two materials in juxtaposition, really, the hard and the soft. The silk also creates a screen, so it can cast a shade onto your actual skin. I wanted to create spaces for you to look and be able to see the actual color mixing that can go on. So a very abstract interpretation, and almost the piece I see as a positivist piece because the boxes become lenses so that you get a layering up and a differentiation of skin color. So you no longer have just one. And genetically, nobody is exactly the same skin color. You could get very, very close within family ranges. The other aspect of this work that I wanted to mention is that I'm fairly interested in modernism, minimalism, and I teach that post-1960s art to my students. And this piece is clearly in reference to the essay, Anna Sheva's essay, Minimalism and the Rhetoric of Power, in which she looks at particularly Donald Judd's work and his writing of specific objects circa 1965. I'm sure if you know Donald Judd's work, you would see that this has a direct reference to his work. And while he's writing that and he's making these very clean, cold pieces with one foot on claiming leftism, a leftist political philosophy, Malcolm X is shot and Vietnam is invaded. And I really wanted to think about this rhetoric that has happened within the art world of being able to stay away from the political. And while Judd's writing these things and other things are going on, there are women and civil rights activists making work, talking about the politics of the day. So, looking at the grid being tossed up into the air. So these pieces, I wanted to have a kind of random, almost jazz-like quality to them in the end. This is the studio, and as I said, the dyeing of the silk and the accessing of it was a really important part of my process. We took the summer, CJ and I learned to dye silk. It was really fun, and learned what different chemical properties do when you put Brazil wood with tannin and alum. It becomes completely black, a solid, beautiful, rich black, to a purple, and then we found an Indian shell, the shell of a bug that turns a beautiful crimson. So this was a really amazing learning process. So I hope that you guys have some questions and found the pieces interesting and see my lineage. I guess visually the work changes over the years, and I hope leaves room for your own interpretation. My hope was that the work could be read certainly as a social and political piece, which it is, but also to be visually engaging. So that's all. done? All right. Yeah.
So you have these rectangular kind of long shapes that you've chosen where the work is being shown. Is that in reference to the genetic testing and kind of the rectangular shape that we get when it's shown the protein levels?
So at first, I was thinking about the categories and boxes that people were being put into. And the grid systems of the color charts were certainly coming up over and over again. And certainly looking at those gene sequencers, they have proteins are clumped into those markers, and it very much like the chromosome 17 piece that I made, right? But I was also thinking about, I won't show you this picture, might bring tears, are orphanages with cribs, just lots of little boxes, right? Housing people, right? And the categories and the confinement of that. So it was really a compelling set of images that came up in my sketchbook. I keep a sketchbook next to me while I do a lot of reading and research. So instead of, I'm not a writer, I'm an artist. So that's kind of how it came about. I had been looking at those kinds of graphics and then thinking about these cages or boxes that things people have been putting into.
Interesting how analogous they are.
Uh-huh. Yeah. Yeah.
Dealing in socio-political art and like having these kind of very hard, like harsh, hard truth topics that you have to deal with in your art. Going to museums stuff, I imagine that like you have a lot of people who are very intellectual and like receptive to this kind of thing. Have you ever met with friction or confrontation with people when you approach anyone or audiences or museums with these kinds of projects?
Not overtly. Probably people are pretty polite in audiences like this. But I would imagine, and probably there should be more dialogue along the way. But certainly asking me if it's real science. And it certainly made me be very aware that I couldn't just use science. And that happened in graduate school. I think my feet were held to the fire enough that I couldn't just decide I was going to make a collector's chamber and not get the right definition of that flower because somebody in the room would know, right? And particularly working with the Department of Medical Genetics, they'd get very upset with me. I'm terribly dyslexic. And I get really mad when I say, I'm sure I said many things incorrectly today, but they would get really mad if I did it incorrectly. So that's mostly what I get in trouble for. But in the broader picture, I've shown you my career of 20-some years, a little bit more, and I certainly have sold my work, and there is a gallery that shows my work in New York. But I have not been engaged in the commercial world of making my work. It's research-based. It takes a long time. So I would imagine that if I was making this and trying to have a sales pitch with it, there might be some controversy, right? So it's been a little bit of a different pace and a different audience that might be interested in looking at that. So, any other questions? Yeah.
Well, you talked about how, obviously you're a teacher as well, and you talked about the importance of your students and how your students give back to you. And I'm wondering if you could just talk a little bit more about that as being a serious practicing of this full-time teaching. How does that work together, and how do your students help you in creating?
Well, it's the questions that they bring to the table and issues that they're wrestling with, I think, that have fueled most of these projects. And because so many students who are in the sciences end up in the sculpture studio, we can make some analogies. They're visual, physical thinkers, but I have a larger proportion in my classes than in other arts classes. And they think in a certain way, and they're working on certain kinds of problems about the structures of life or they bring in the DNA fingerprint, and we're reading Walter Benjamin, Art in the Age of Mechanical Reproduction, and they say, I have a DNA fingerprint, check this out. Here's all new, and they do it for their little visual concepts project or for their, sculpture project. And it's inspired me. And sometimes I then hire them to work with me in the summers. So there's that. But it's also, being at Reed, they're really critical thinkers, and they hold my feet to the fire. So I have to come to the table responsible for what I'm about to say. So I think it's made me want to make work in a slower pace and with research behind it, so it's not, yeah. Yeah. It's a different audience, I think. Yeah. Jackie. I know the audience here.
Could you talk a little bit about the STAR project and in particular the reception of the STAR?
Yeah, well, that's an interesting question because I was just asked about the controversy, and we have a very close friend who I brought a housewarming present that I wanted to make them a blanket from one of the chromosome markers, and they didn't want it because it had to do with cancer. And those who have been interested in supporting it often have some history of survivors. My own mom died of cancer, and I've had many people around me be survivors, and I know how important it is to have help. And so the physicality of that was what I was after. But other people find it abject to wear something that says breast cancer on it, even if it's beautiful. So it's been interesting. Yeah. And I also understand the controversy with it. Actually, Carla Mann and I had a really amazing conversation over the film, The Pink Ribbon, that's about the breast cancer industry. So there's a whole corporate connection to supporting raising money for breast cancer. And many of those corporations are corporations that produce untold amounts of pollutants that are probably causing breast cancer. So plastics, for example. The water bottle company, Dance, is one of the big culprits. So it's a really hard play. So I know about wanting to sell things to raise money for cancer. It's also pulling at the heartstrings of people who are already, suffering. So that's partly why I just did 10. I did it. I'm not doing it anymore. I'm done. I also had a little toe. I have a friend who's in the textile industry who kind of helped me along the way, figuring out what size should these be, and what would be the best way to produce these. And for a while, I thought, oh wow, we could get a sponsorship or something. And I looked into that, and it was just unbelievable. If you would try to take them to Macy's or something. I never tried to do this, but you'd have to have them printed in China. I made them all in the US. I sewed them with former students. So they're, totally homespun. But I had to make them so that they looked pretty sellable. But yeah, it was a very controversial part of that. I didn't want to go beyond the 10. I didn't want to go to a corporate sponsorship because then it became this big animal that I teach for a living. I don't have time for that. Yeah. Anyway. Yes, Chris.
I was really struck by how persistently your work thinks about visual representations of science, not just science per se. And so I'm just trying to, I'm asking you if you remember, is there a piece that you showed us or that you didn't show us in your past that deals with that produces a kind of physical manifestation of something that doesn't already have a visual manifestation native to science? In other words, is there something that you ever produced that is sort of invisible prior to you making a manifestation of it? Or is there always a kind of scientific mode of representation that you then turn?
And I think what's interesting about that question is that I'm interested in the chromosome paintings is the geneticists making something visual that wasn't visual. So I'm using their visualization for sure. A piece I didn't show was a sound waves pattern. I recorded the bird songs. This is before the cellular pieces, sort of between the plant pieces. And I carved them on long wood blocks. I might be able to show you, but the piece had both an auditory and the visualization of sound wave patterns in it. But that is a motivation. I'm trying to find, like, who are the smart people out there that have those visualizations so I can make a representation of them. Yeah. Yes?
I have a question about the division of the cancerous cells versus the healthy ones. Are those two separate samples that were tested?
Yeah. The piece that I made, Andrew Pelling gave me 5 soundtracks, and they're different phases of cell division and different levels of health or illness that they had. So cells that were terribly diseased, right, to perfectly healthy. And he gave me only 5 and gave me copyright to use them, so I used them to make this piece. And I tried to make the sounds, I tried to orchestrate them. The video's online as well. But yeah, very distinct, separate recordings of only 3 or 4 minutes long each. So they're repeating as well. I learned to use Sound Mixer to do that. Yeah. Any other questions? Yeah, you could go see the piece and you could ask me questions there about making the piece or whatever. Thank you all for coming on a Saturday.